Mm. Oh, hey there. My name is Caleb Cease, and welcome to this episode of Sick Genes. So, personally, I'm not a big fan of modern moral and ethical dilemmas, but that's exactly what we're going to be talking about today. Are you sick and tired of the way you look? Personally, I'm so tired of being self-absorbed, like all my other little science friends tell me. Anyway, do you want super babies? I mean, don't we all? Everyone has NBA babies, million-point games. It would be great. That's besides the point. Well, now, with modern gene editing, you can change the way you look and have NBA super babies. Let me point this over to my friend Jacob Webb to talk about CRISPR Cas9.
Hello, I'm Dr. Webb, and the first type of gene editing we are going to talk about today is clustered regularly interspaced short palindromic repeats, or also known as CRISPR Cas9. This system is regularly found in bacteria, and there are two parts to this CRISPR system. The first part is the Cas enzyme that breaks out the needed DNA sequence. Then there is the guide RNA that is used to locate the targeted genetic material for the Cas enzyme, so the RNA basically works with the Cas protein to locate the specific DNA sequence that needs to be cut out. Once this process has taken place, the cut in the DNA now leaves room to repair the gene or replace it, and that is your simple breakdown of CRISPR Cas9.
Oh, wow. Funny seeing you here again. Well, thank you, Dr. Webb. That was mighty sciencey. Let's pass it on over to Dr. O'Brien for the second gene editing system. Thank you.
Hello, I'm Dr. O'Brien, and another type of gene editing is called transcription activator-like effector nucleases, also known as Talons. Talons are chimeric proteins that contain two functional domains, a DNA recognition transcription activator-like effector, and a nuclease domain. They work for gene editing by recognizing a specific sequence which the user can design and introduce a double-stranded break with an overhang. The main difference between CRISPR and Talons is that CRISPR can cause multiple gene mutations with one injection, while Talons are limited to one simple mutation. However, a study conducted by researchers at University of Illinois found that Talons was up to 5 times more efficient than CRISPR Cas9. So what are the benefits to taking Talons gene editing? Well, one benefit is that Talons system uses simple and easily predictable code, which makes it extremely precise in its works.
G, thank you for joining me for Evening Espresso and Research Time. A special thanks to my two friends for explaining the two main gene editing techniques. Now, that might be a little complicated for the average person to understand. Well, I wrote up this little article, so maybe the average person can understand gene editing a little better. So here we go. So what exactly is all this gene editing mess? The main goal of gene editing is to either take out, add in, or change genes entirely. The motives behind can stem from disease, appearance, or genetic abnormality. Gene editing takes proteins and changes the way they are produced in the cell. With this technology, it opens many, and I mean many, ethical and moral dilemmas. Well, that still might not be clear enough. So let me pass it to my favorite doctor, Dr. Rob, over in the lab to give you an actual representation of CRISPR Cas9 and Talons. Thank you.
Hi there, I'm Dr. Rob, and welcome to my gene lab. I'm going to talk to you today about how gene editing really works. In simple words, gene editing is a group of technologies that allow scientists the ability to change an organism's DNA. Material can be added, removed, or altered in a particular location in the genome. For example, recent studies done to use pig organs as an efficient alternative for human transplants. Scientists have used CRISPR to remove viruses from pig DNA to produce a clean, safe, virus-free organ for possible human use. Research is still being done to make this an available option. However, we can see this being a serious possibility in the future. Now, if you step into my gene lab, you can see that we have this worn, old pair of genes here, while we also have this fresh, nice pair of genes. However, a slight Sharpie mark in my nice, fresh pair of genes. Now, with a little bit of gene editing, we can take the genes and create a fresh pair of genes. Perfect.